Guys, and welcome back to another video. Today we're going to be looking at receptors and how they work, and then we might go, yes, we will go into a case study on the Bacillian corpuscle receptor. Okay, so receptors detect stimuli. That's the main reason for their existence, and they pass information about this stimuli along a nervous pathway to the CNS. And we need to know how they work. So, receptors are specific, and obviously I previously mentioned the Bacillian corpuscle as a specific receptor for vibrational pressure. Receptors like light receptors, we've got the rod and cone cells, for example. There are many different types of receptors that each detect a different type of stimulus. Some receptors are cells, like photoreceptors, and some receptors are proteins that are on cell surface membranes, like glucose receptors in some pancreatic cells. So, receptors in the nervous system convert energy of the stimulus into the electrical energy used by neurons, and this is how they work. So, as you can see the diagram here, we're going to be going through resting potential, threshold potential, generator potential, and the resting potential. When a nervous system receptor is in its resting state, i.e. not being stimulated, there's a difference in charge between the inside and the outside of the cell, and the inside is negatively charged relative to the outside. This means there's a voltage across the membrane. Now, it's important to note that both cells are positively charged with positive ions. However, the outside of the cell has more ions, so is more positively charged than the inside of the cell, hence meaning the inside of the cell is negatively charged. Okay, and it's obviously relative. So the voltage across the membrane is known as the potential difference. And the potential difference, as you can see on the y-axis, or the membrane potential, when a cell is at rest, is called resting potential. And we can see that resting potential on the graph is roughly negative 65. And the cell is always, when non-stimulated, at that negative 65 point, as we can see, resting potential. The resting potential is created and maintained by sodium-potassium pumps and potassium ion channels in a neuron's membrane. Sodium-potassium pumps use active transport to move 3 sodium ions out of the neuron for every 2 potassium ions that are moved in. ATP is needed to do this. Now it's important to note that a sodium ion is written as Na superscript plus, and potassium ion is written as K superscript plus. Okay, so potassium ion channels allow facilitated diffusion of potassium ions out of the neuron down their concentration gradient. So that means overall more ions are getting pumped outside of the cell than inside, which maintains that resting potential gradient that we want. Okay, and also it's important to note that the sodium-potassium pumps move sodium ions out of the neuron, but the membrane isn't permeable to sodium ions, so they can't diffuse back in. And this creates a sodium-ion electrochemical gradient, because there are more positive sodium ions outside of the cell than inside. And that's basically what I said earlier. The sodium-potassium pumps also move potassium ions into the neuron, and that just further balances out the gradient. And when the cell's at rest, most potassium ion channels are open, and this means that the membrane is permeable to potassium ions, so some diffuse back out through potassium ion channels, and obviously not using active transport as it's facilitated diffusion. Okay, so then let's move on to the action potentials. So, when a neuron is stimulated, other ions in the cell membrane called sodium ion channels open. If the stimulus is big enough, it will trigger a rapid change in potential difference. And this causes the cell to become depolarized, as it's no longer polarized, which it is previously at resting potential. And this sequence of events that happens is known as the action potential. Now, if we look at the figure, we can see the action potential, which is when it goes and reaches the threshold and moves from about negative 50 all the way up to positive 35. So let's talk that through. So obviously a stimulus excites the neuron cell membrane, causing the sodium channel ion channels to open, and this membrane becomes more permeable to sodium, so sodium ion diffuses into the neuron down its sodium ion electrochemical gradient. And this makes the inside of the neuron less negative. So then we obviously have depolarization. If the potential difference reaches a threshold of around -55 that we can see here, more sodium ion channels will open and more ions will diffuse into the neuron. So then we will have a depolarization, and then we will have repolarization. So at the potential difference of around +30 to 35, the sodium ion channels close and these things called potassium ion channels open. The membrane is more permeable to potassium, so potassium ions diffuse out of the neuron down the potassium ion concentration gradient. And this starts to get the membrane back to its resting potential. However, the cell does not return to the resting potential without doing hyperpolarization. And hyperpolarization is when potassium ion channels are too slow to close. So there's a slight overshoot where too many potassium ions diffuse out of the neuron. And this potential difference becomes more negative than the resting potential, which in this instance is -65, so it might become -70 or -75. And then eventually the ion channels are reset and the cell returns back to the resting potential. The sodium-potassium pump returns the membrane to its resting potential by pumping sodium ions out and potassium ions in, and this maintains the resting potential until the membrane is stimulated once again. So it's important to also note that action potentials are all one size, so the strength of the stimulus is measured by the frequency of action potentials, and the brain picks up the gap between them. The number of, so if the stimulus is too weak, the generated potential won't reach threshold, and so there'll be no action potential at all. It's very important to note that if the stimulus is not reached, no matter how close it is, so it's 1 millivolt off, it will not reach action potential. Let's also go over the refractory period, which does include hyperpolarization, which we've already mentioned. So after an action potential, the neuron cell membrane cannot be excited again straight away, and this is because the ion channels are recovering and they can't be made to be open. Sodium ion channels are closed during repolarization, and potassium ion channels are closed during hyperpolarization, and this period is known as the refractory period, as I just previously mentioned. And so then we'll talk about waves of depolarization. And when an action potential happens, some of the sodium ions that enter the neuron diffuse sideways, and this causes sodium ion channels in the next region of the neuron to open, and sodium ions diffuse into that part, and this causes a wave of depolarization to travel along the neuron, and this wave moves away from the parts of the membrane in refractory period because these parts can't be stimulated to fire an action potential. So it's almost like a Mexican wave, and it's very interesting. We could talk about saltatory conduction and speed of conduction, but I think I'll put that in the next video, and we'll do like a part 1, part 2 type thing. So I'm going to wrap it up here, and thank you for watching, and I will see you in the next one.